This is Dr. Meena Rao, Associate Editor for Physiological Reports. Today we will be discussing, quote, leaky intestine and impaired microbiome in an amyotropic lateral sclerosis mouse model with Dr. Jun Sun. She's a lead investigator on the paper authored by Xiaoping Wu, Jianjun Yi, Yongguo Zhang, Jinsong Zhu, and herself that appeared in Physiological Reports in March of 2015. This stimulating paper reported for the first time a potential and novel role of the small and large intestinal epithelia and the gut microbiome in the progression of the neurodegenerative disorder amyotropic lateral sclerosis. Dr. Sun, thank you for being here. Could you please briefly describe your exciting work on ALS?
Thank you, Dr. Rao. ALS is defined as a motor neuron disease because of its pathogenic hallmark is the death of the motor neurons in the spinal cord and the brain. For the first time, our study discovered a link between intestinal homeostasis and the microbiome in ALS progression. We examined the gut of an ALS mouse model called G93A at the age before the ALS symptom onset. We have discovered damaged tight junction proteins with the increased intestinal permeability, which is called the leaky gut. Furthermore, we have identified some increased abnormal Paneth cells in the small intestine of this ALS mouse. What is Paneth cells? Paneth cells are specialized intestinal epithelial cells that sense the microbiome in the intestinal GI tract. So we further identified by sequencing the intestinal microbiome, we clearly see the shift of the profile of microbiome in the ALS mouse model compared to the wild type mice. So in this way, we identified specific change of G93A mice showed abnormal microbiome and also the dysfunction of GI functions like gut-barrier functions. And furthermore, we identified some butyrate producing bacteria which is disappeared or lacked of expression in the ALS mouse model. So it's quite exciting.
So that is really a very exciting discovery, and it's unusual to make the connection between neurodegenerative diseases and gut. So scientific discovery is often serendipity. So what really made you look for a connection between ALS and gut biology?
That's a great question. I was asked by these questions many times because GI is considered as the second brain of the human being because it contains roughly the same amount of neurons as the spinal cord. And in the meantime, we have noticed that actually commensal bacteria outnumbers over human cells by almost 10-fold in the GI. So recently study, we have already found evidence that the microbiome and intestinal homeostasis contributes to neurodegeneration diseases such as autism, Parkinson's disease. However, very little is linked between the GI crosstalk with the ALS. This is the theory because we're trying to test the concept where the microbiome and the intestine really contribute to ALS. And in the real life, you need to find a collaborator who is willing to test your theory. So I'm very lucky to have my collaborator, Dr. Jinsong Zhou. She's also the co-corresponding author in this paper. And she's willing to test my hypothesis with her animal model. So we work together using the ALS mice. Before the onset of the disease, we're able to find a difference of the microbiome and also the intestinal functions in these mice compared to the wild type.
Great. So the whole notion, therefore, of, as you mentioned, of the microbiome influencing or interacting with neurodegenerative disorders is a relatively recent concept, right?
Exactly.
And so what do you think comes first, the microbial dysbiosis or the neurodegenerative disorder?
Because this is a very nice question about which comes first, chicken or egg, right?
Right.
And you can easily imagine if this disease happens at the onset of the disease, the early stage, before you can see any ALS symptom, you already identified the leaky gut, the dysbiosis, which is the imbalance of the microbiome, which gave us some clue. The GI could contribute partially to the pathogenesis of ALS. We have to say that in a very conservative way because we haven't really identified further what's the time course, what's the progression of the disease along with the GI dysfunction. At this stage, we cannot clearly say, okay, is this the cause of the GI, problem lead to the ALS? But I would say it will contribute to the onset or the early pathogenesis of the disease based on our observation in the mouse model.
Yeah, especially because you were able to see these changes in the gut in the mouse model, before the onset, the neurodegeneration of the motor neurons.
Exactly.
So there's
some indication. Yeah.
Great. So we can probably spend the whole afternoon talking about this.
Yeah.
But I was wondering if you could tell us what are the next steps of this exciting research, both from a basic science perspective and in terms of the clinical implications of your findings.
Yeah, for the basic research, based on our reports, we have already submitted NIH R1, which trying to understand the mechanism behind the gut and the neuromuscular crosstalk, especially the mutation, which is called SOD1 mutation in these mice. We want to understand the mechanism.
So SOD being superoxide dismutase.
Exactly.
Right?
Yeah, it has a mutation in this ALS mouse model. Another interesting part is we actually have gotten many inquiries from the ALS scientific community and also from ALS patients. This study is also further funded by ALS Association Pilot Award. They announced us to study what's the potential to target microbiome. Will that strategy eliminate the progression of ALS? And in the meantime, we got some response from the patient because they were told because maybe GI is not a big deal in the ALS progression, but they actually noticed some GI problem even before the onset of the disease. Because we're talking about the potential application. One is use the GI symptom as a marker
for ALS
diagnosis, which is a challenge in the field. Another possibility is can we really targeting the GI functions, like also targeting the microbiome, develop some novel strategy for ALS treatment. Two directions to go, I think, for the application.
That's really exciting, and it actually brings us back to the question about the serendipitous observations, right? You didn't quite touch upon whether you and Dr. Zhou decided this over a water cooler conversation, that she had the model and you had the methodology for gut microbiome, but also the huge involvement, and I think the patients got really excited by your study from what you've told me, because they read your article in Physiological Reports, right?
Exactly.
And it's hit the press, so that's really exciting. And, and patients very often will have these anecdotal observations that they're the ones who know about gut dysfunction, and maybe the doctors never made that connection. So this is potentially very, very exciting.
Exactly, and I really appreciate Physiological Reports reported our study.
Right.
Thank you very much.
Right. Well, thank you. Thank you for discussing this important study with us, and we really look forward to seeing your future work in the journal, and this has been very exciting.